Okay, so last week we have discussed a little bit about the structure of cells. Yeah, why we studied the cells, because the cell is the basic structural, the basic structure, yeah, the basic level structure within our body. All of the organs are made from tissues, and all tissues are made from organization of cells. The cells are organized, they are assembled, and uh, they become arranged or aligned with other cells, and then they make tissue. So most of or many of the physiological phenomena basically starts from the cell, yeah, not from the other. Some others may start at the higher level, but um, many major physiological phenomena starts from the cell. Moment, let me pause the record. So that is why the knowledge about the cells is important to understand how the some physiological phenomena occurs or some biosignals is generated. And even at the advanced level, the recording of the biosignal can be done also at the cellular level, yeah, to know exactly at where. Actually, that is one of the main bottleneck now with most of the current biomedical instrumentation, however, still record the biosignal at the individual level or at the organ level. It is not yet too feasible to extract information at the cellular level, unless if the cell or the tissue is being biopsied, with a procedure called biopsy, being cut, being taken away, and then cultured first at the laboratory, and then being investigated for its physiology. For example, in the case of cancer diagnostic, cervical cancer diagnostic is done by pap smear test, using pap smear test, and then the tissue taken from the cervix is being uh, cultured in the lab to see the morphology. Another cancer diagnostic, like breast cancer, for example, also done first after the mammography to get an early indication whether certain part of the tissue in the breast is tumorous or cancerous or not. After that, for confirmation, the doctor will still need to do biopsy, take some sample, and then culture in the lab, in the pathohistology lab. And then based on the morphology, they will determine whether it is normal tissue or cancerous tissue, or then how far the stage is already. So yeah, most of these are done at the cellular level. Some, like electrocardiography, on the other hand, later we will study how it is generated. We can only measure for now at the organ level, yeah, or the individual level, as the electrical response coming from the heart tissue. While actually the the signal itself may start from individual cells within the heart cells, but so far there is no convenient technique that we can measure or we can localize the cause of the particular heart signal at the cellular level. So that is the current current condition of biological biosignal measurement at the cellular level. Nevertheless, we still need to study the cell functional structure, and we have discussed about this last week. And we have covered a little bit, we've introduced a little bit the most important molecule that form the cellular membrane. Why do we study cellular membrane? Because at the membrane is what the ions transfer usually occurs. And this ion transfer is what is responsible for the occurrence of biosignals at the higher level. So we have studied the cell membrane phospholipid biomolecule, which basically is formed from the hydrophilic head, consisting of glycerol, choline, and phosphate functional group, and the fatty acid hydrophobic tails. And in the cellular membrane, the fatty acids will bind with the other fatty acids at the other layer of the cell, the other layer of the cellular membrane, while the hydrophilic head will be facing the aqueous face. Yeah, in this case, in the outer side, it will face the extracellular liquid. At the inner cell, it will face the cell plasma. Okay, so we see that why does that happen? That thing happen because the hydrophilic part has parts, yeah, has functional groups which are very polar. Yeah. You remember polar in chemistry. Yeah, also we cover a little bit in the biomaterial. So the polar part of the biomolecule will bind easily to the polar molecules of water in the surrounding water. On the other hand, the hydrophobic part, it is not polar at all. It is non-polar. So it will bind easily also to the non-polar part of the lipid molecule, yeah, in the cellular bilayer lipid membrane. So what occurs is there is an electrostatic force, yeah, between the attracting structure, yeah, between the polar structure here, yeah, between the polar hydrophilic head of the lipid molecule with the surrounding water molecule. And this is formalized with this formula. Yeah, W is the energy of the attraction equals to the integral of the electric field times the electric flux density D divided by 2 integral over a certain volume. And this is equals to d square integral of our volume, d square over 2 epsilon. Epsilon is the permittivity of the medium. Besides the lipid membrane, the lipid molecule itself, the membrane also contains protein. So the protein could be a protein only, it can be also an enzyme, it could be also an antigen, it could also be linked, yeah, conjugated, we call it, to carbohydrates, and we call it as glycoprotein. And these glycoproteins are responsible for giving negative charges to the outer surface of the cell. So the bonding between biomolecules occurs if these two conditions are fulfilled. We have covered also last week. First, they have to have complementary charges. If they are polar, then the plus will bind to the negative part of the other molecules. And the shapes also has to be complementary. Yeah, this is the lock and key concept, which you learn in biochemistry. So something like this. This can only bind this if the shape is complementary and the charges are also opposite to each other. The function of the enzyme that in that case will be to facilitate where they can bind faster. Yeah, so here the enzyme has also a complementary structure that can that complement the shape of the binding, the bond molecule between the two molecules. Yeah. So cell membrane, all cells has electric voltage U across their membranes. The resting state U equals to UR, yeah, where UR is the resting voltage amplitude. Some cells are electrically excitable. For example, the nerve cells in brain, the muscle cells in muscle, yeah, using an impulse U. So that is why there is a technique called electric injection tomography for imaging. There is also technical electrical injection therapy, where current is being injected into the cells, into the nerve cells or the muscle cells to stimulate that current. For example, in the muscle cells, then in order to enable training of the muscles without the person itself has to consciously move that particular parts. For example, some muscles like the stomach muscle. Yeah? How how can we have exercise of the stomach muscle even while we are not in the exercise condition. So there are some devices in the market that can be put like a pad on above this stomach muscles, and then that device will inject some electrical current to excite the muscles so that it will contract and then relax, contract and relax to simulate an exercise. Similarly, the brain can also be excited using this electrical current. Some therapy uses this method, although not yet widely available. So the UT will carry many physiological information, UT from the cell. That is the cellular response to that stimulus. The electric membrane behavior will determine the origin of all the biosignal. See, because in the membrane itself, ions crosses through, okay, and the difference between the potential inside the cells and outside the membrane, outside the cells, bordered by the membrane, determines this condition of the biosignal. And that is why then cell membrane has two types of properties. First is the passive behavior, reflecting the sustained properties of the membrane, and then the active behavior. The active behavior comes from the regulatory mechanism through the gated ion electrodes, as well as the active transport. The active transport is transport of substances through the diffusion gradient across the membrane. The regulatory mechanism occurs through the gated ion electrode, where the ion electrodes pass through ions using gate molecule that will close or open on that ion channel. Active transport, on the other hand, uses the diffusion gradient, as well as it can also use an active energy from ATP, adenosine triphosphate of the cell, to transport the ion actively. So this is the passive properties. So we have ion channel, passive channel, protein. In this case, this is for potassium plus ion, K plus ion, which is normally located. There are more number of potassium ions inside the space, inside the cell, inside in the intracellular space. So the resting non-gated passive channel protein for potassium ion transversing the cell membrane. In this case, the channel is always open and govern the resting voltage amplitude UR across the membrane in the resting state of the cell. Within the channel, the net transport of potassium plus ion across the membrane is governed by an equilibrium between the diffusional force Fd. The Fd will push the potassium ion from the intracellular space into the extracellular space, which is diffusion based. Diffusion based, we will learn later in a biomaterial. When the concentration of the potassium ion is higher here and lower at the extracellular space, so the potassium ion will tend to travel from the higher concentration space into the lower concentration space. Okay. So this is called the FD, the diffusional force. However, when it starts to move upward towards the extracellular space, one by one move, then it will start to bind electrical field. Okay. The electrical field will push in the opposite direction. Why is that? So because here you see that when more positive potassium ion move towards the extracellular space, the positive charge is built up on the extracellular membrane. Okay. So, and the negative charge will build on the intracellular space membrane. So there will be electric field from the positive to the negative, and this will be in the opposite direction of the movement of the potassium ion charge. Okay, so the movement, the diffusion of the potassium charge will occurs until this electric field is in balance with the diffusional force. So due to this motion also we have relative potential difference between the outside of the membrane and the insides of the membrane. So this is the experimental arrangement to detect passive electrical behavior of the membrane. So you have here the microelectrode, you have here the ground electrode, the potential, and this is the intracellular space. So this is an intracellular stimulation of the action of a nerve cell in order to attain characteristic data on passive electrical behavior of the membrane of the axon. Okay. So the step current I is applied. This is applied using a step current. Step current means from 0 it is being pulled to the one stage, from off to on stage, injecting current here, the electrons into the inner space of the cell. Then it is simultaneously registered. The voltage, the occurred voltage is registered by this voltmeter with simultaneous registration of the resulting membrane voltage U across the membrane, this membrane voltage U, at the distance x from the stimulation side. The distance x is here, from the distance from the recording probe from the injecting probe. This is the distance. The induced currents flow from the inserted microelectrode into the intracellular medium across the membrane outwards, enter the extracellular medium, and then return to the current source. Return to the current source here at the ground. The arrow's thickness indicates this thickness, local strength of the induced current density. So for example, here, the induced current density, of course, near the injecting probe is larger compared to the distance farther away from this injecting probe, injecting needle. So this is one example where we inject current here at position 1, 10 picoampere per milliseconds, and then position 2 measure the resulting voltage. Okay. So this is similar. This is the current, injecting current. This is the resulting. The blue one is the resulting voltage. So this is called the microneedle technique to investigate the property of a cell based on the resulting current, resulting voltage from an intracellular current stimulation. Okay, so this is to know the passive electrical behavior of the membrane. The passive property which results from this open channel, that makes where ions, positive or negative, can flow freely across that channel. So for example, in this picture, the positive ion of potassium travels from the intracellular space into the extracellular space. The chlorine ion flows from extracellular space into the intracellular space. The sodium plus ion also flows from the extracellular space into the intracellular space. So using this technique, we can know that balance or equilibrium condition of the ions within the particular cell. But it is not specific to which ion. In how to know in active mechanism, There is a regulatory mechanism in the membrane which gate the channel protein, and there is also active transport of ionic substances up their diffusion gradient. What does it mean with up their diffusion gradient? Normally, the ion will be transported down the diffusion gradient. It means from the higher concentration to the lower concentration. In the active transport of ionic substances is the cell tries to pump the ion from the lower concentration into the higher concentration of that ion. And that is what we call as active transport, which requires the use of the cellular energy, which is the ATP, adenosine triphosphate. So this one is the regulatory mechanism. The first one is voltage gated ion channel. Yeah, in voltage gated ion channel, for example, here, this is when the channel is being closed by this gate. The negative charge of the activation gate. This is closed, so the sodium ion cannot travel into the intracellular space. Then, when the charge here becomes negative, so this is now positive and this is negative. When the charge is reversed, this is negative and this is positive. Yeah. So this negatively charged gate will be attracted by the positive charge of this intracellular space. Yeah, this will goes towards the intracellular space and then makes the channel open, so that then the sodium positive ion can travel from the extracellular space into the intracellular space. Okay. If here is the condition U equal to UR, here the UR is very much less than U. In the transmitter gated, so this is voltage, yeah, gated by the voltage difference. So here, when the extracellular space has higher potential compared to the intracellular space, the gate is closed. When it is smaller than the intracellular space, the gate is open. UR is smaller than U, then the gate is open. Transmitter gate, on the other hand, opens when there is a receptor site at the ion channel, which will bind to transmitter. Transmitter here could be any molecule, yeah, that responsible for the opening or closing of gate. An example will be like acetylcholine, something like that, or many other molecules later. Some hormone. This could be the transmitter that will open or close gate. So, for example, in this case, when the transmitter is not binding to the receptor, then it is closed. Okay, so it is in the resting state. Yeah. But when the transmitter bind to the receptor of the ion channel, then the gate will start to open. So that then the ion can start to cross and traversing the channel. So far, clear? Any questions regarding the active and the passive mechanism? So this is the voltage gated channel as what we have discussed here. Yeah, this is the cell membrane. So at normal condition, when the voltage is negative 70 millivolt, yeah, this one is negative 70 millivolt compared to the upper part. There is this molecule that will bind. The channel will be closed by this gate. But when there is the voltage is becomes higher, and becomes higher means that minus 50 millivolt is larger, right, than minus 70. So this will open, and then these ions can start to flow through the channel, going out to the extracellular fluid. So this is voltage gated channel. In the transducer gated, it requires another hormone or molecule to bind to the receptor part of the channel, so that when upon binding, the conformation structure of the channel will start to change. It will move, and then the gate will open. So like this is a neurotransmitter. The ligand is required to open the ion channel. In this case, is ACH. ACH is acetylcholine. Yeah. So when it is not binding yet, all these ions, the sodium plus, the calcium plus, the potassium, cannot go to the other side of the membrane. Then, when it binds, the neurotransmitter attached to the receptor, and this chemical stimulus will open the channel. Yeah, and then potassium can flow from inner cell to the extracellular liquid. Sodium and calcium can flow from the extracellular liquid to the inner cell. So, of course, this is still gradient diffusion control. It means that the flow direction will be dependent on the concentration gradient. In this case, in this particular picture, since sodium concentration is higher at the extracellular fluid, so when the channel opens, it is flowing from the extracellular fluid into the cytosol. Potassium case, since the cytosol concentration is higher compared to the extracellular fluid, so when the channel opens, it will flow to the extracellular fluid. So this is the active transport. In the active transport case, when active transport case is different than this one. This is passive, active, but based on regulatory mechanism. Either this is transmitter gate, this is voltage gate. Okay. In the active transport, the ion move opposite to the concentration gradient. Okay. So even though here in this case that potassium, or let's see which one, the sodium ion, yeah, the sodium ion is higher in the extracellular space compared to the intracellular space. Here we would like to transport the sodium from the intracellular space into the outside, into the extracellular space. So here when the channel opens, this requires this ATP, adenosine triphosphate, to boost the energy, yeah, for the sodium ion to be able to move outside, where actually it has higher concentration of sodium. And so actually it's like moving water from lower level to higher level. Actually it is not natural, but you can do that using pump. So this ATP acts as the molecular pump inside this channel. The ions are transported against their diffusion gradient, requiring the hydrolysis of adenosine triphosphate, ATP, to provide the necessary energy. Binding of 3 sodium ion here binds in the inner wall of the channel and releasing 2 positive ion inside the cell. And B, the subsequent release of the 3 sodium ion and binding of these 2 new potassium ion outside the cell. Yeah, so the potassium is being pumped from extracellular space into the intracellular space, into the cytosol. The sodium is being pumped from the cytosol into the extracellular medium. Okay, so far is clear regarding the transport of ions across the membrane. Any questions? Okay, if not, we continue here on measuring the cell membrane potential. Finish here quiescent cells and excited cell. The excited cell is using cell stimulation, cell response, and respond to the different stimuli. This is the model Quiescent cell means that when it is at resting state, resting potential, the model is based on resistor and a voltage source here. Yeah, this one represents the voltage source, represents the or modeling the ion, the voltage caused by each of the ion type within the cell. Yeah, there is this potassium, sodium, and chlorine, and then there is a capacity current across the membrane. Then, if we have action potential, postsynaptic potential, and the resistor is the conductance. Yeah, here is model as variable conductance, the G of the potassium, G of the sodium, and G of the chlorine. This one is what earlier being presented in the earlier slide, that we inject a current, stimulating the cell with a current, and then registering the response, the voltage response potential using another needle, okay, and which will be then a function of the space as well. The equivalent circuit model is like this. This is the induced current. The induced current then will flow across this model of the cell, yeah, like previously drawn here, consisting of a voltage source as well as a conductance, with the conductors which will depend also on the distance to the recording electrode, as well as then there is also capacitance here. So the response of which, when we excite the cell using an excitatory current stimulus, a step current stimulus, yeah, then the response will be something like that. It will cause depolarization of the cell membrane. Depolarization means from this very negative potential difference, it will go towards the zero, yeah, and then go toward higher potential. After that, repolarization again. When the step finishes like this, it starts to repolarize again as the normal condition at the resting level. So based on, for example, how high is the depolarization, what is the time taken from this depolarization state until it goes to the resting level again, that will determine, from there we can determine the state of the cell. In this excited cell, action membrane potential, in this A, voltage U across the cell membrane as an action impulse. So we excite a voltage U in millivolt, action impulse. Then the corresponding behavior of the area related membrane conductance, G dash for potassium and G dash double dash sodium for potassium and sodium plus ions respectively, and see the equivalent circuit model in figure 2.15. So this is the equivalent circuit model. This one. So it has basically two ion channels which we want to model, the potassium and the sodium. Okay. So this is the influx of the sodium plus, and this is the efflux. Influx means coming in from the extracellular plasma into the cytosol, where efflux of potassium means that the potassium ion is going out from the cytosol into the extracellular plasma. The C here is the corresponding ionic component, Je, and capacitive component, Jc, of the total current density here. J is the total current density in milliampere per square centimeter through the membrane. Typical absolute values of U, G, double dash, and J are given in each case with the data being partially taken. So this one. So this is the model. Yeah, usually we model it as conductance, variable conductance. Why is variable conductance? Yeah, when we have an action potential, yeah, not just resting, but we excite it with certain potential, so the conductance will change. Remember that we have the voltage gated ion channel, which the channel which opens or closes based upon the potential difference between the two sides of the membrane. So this is a various response of cells to the different stimuli, whether the stimuli is given as a step like this, yeah, although in reality it is not ideal. Usually it will need some time, what we call here as TR, as the response time, until it fully goes to the steady state value. Yeah, or likewise also when it goes to the zero state, also it takes some time. Yeah. The response will be something like this. It will cause some decay, and this is caused by the capacitive current of the cell. When we put a stimuli in the form of sawtooth waveform like this, yeah, triangular waveform with amplitude I, with period T, this is what will occur. Yeah, so the capacitive current like this will decay, but upon which then after certain value it will go up again because accommodation state. When we excite with sinusoidal stimuli like this, this is like combination of this. Yeah. First is like this, accommodation, and then going up again. This is how the response will look like in the excited cell. So using this pattern of excitation, stimulus, and the response, then we can study how sensory neuron behave. So in this case, for example, this is neuron which is stimulated by mechanical stimulus. So this neuron, the sensory neuron, one of its end, yeah, is connected to the muscles. So where upon the muscular stretching, so this will excite, it will bind, will mix some of the channel open and close, and then will excite this stretching will excite this potential amplitude. Okay, so the receptor potential at this end will look like this, and then at this part it will create this kind of pulses. When it is above certain threshold, then it will create pulses with certain frequency. And in this end, where it is connected to another part, this is connected to motoric neuron. Motoric neuron, on the other hand, is excited by the signal coming from the nervous system. Yeah, like this action potential, and this action potential is also propagated like this before it goes to the muscles, yeah, which will go through a transformation from relaxed state into contracted state. So this is how the information flow happens. Yeah? The direction of the information flow from the sensory ending of the sensory neuron goes to the sensory neuron, and then this is in the what do you call it, reflex state. So not going through the normal central nervous system, but immediately goes to the motoric neuron. The sequence of mechanical stimulus, a sensory neuron with its sensorial ending in the muscle spindle, a motor neuron, and an innervated muscle that constitute a reflex loop during a reflex loop. A mechanical stretch stimulus has a rectangular stretch shape, whereas the corresponding membrane voltage U, yeah, at different side of both neurons shows specific waveform. The stretch sensitive region of the sensory neuron continues into the trigger zone. This is the trigger zone where action potential are generated. After saltatory conduction of the action potential along the myelinated action, where is it? Myelinated action, this one. These potential are passed to the motor neuron over the neuronal synapse. This is the neuronal synapse, the connection between two different neuronal cells. The motor neuron controls the contraction of muscle cells over the neuromuscular synapse. How excitation propagates? This is the conduction of excitation so along the unmyelinated and myelinated action regions. So A, this is the excited depolarized region at the location x equals 0. So this is a excited region. This is unmyelinated non-excited region. So myelination is this one here. This one is unmyelinated region. This is myelinated region. So the myelinated region will occur like this. There is a region of highly positive and negative on the other side of the membrane. Then he shows the spatial course of the voltage U across the membrane that has propagated from the myelinated region into the unmyelinated region. The wave travels. Then at the synapse, the connection between two neuron cell, the voltage potential is propagated using this ions and transmitter molecule like this. Yeah, this transmitter molecules is being put into the vesicle. Remember what vesicle is? Like bilayer lipid membrane structure, but inside the cells. So when it meets with the membrane, the content inside that vesicle will be released. Yeah, this content usually transmitter molecule. Remember, then which will then bind upon the transmitter gated ion channel. Yeah, which will then cause the ion channel on the next neuron to open or close, and then cause the ions to be able to be exchanged, yeah, between the neuron and the surrounding extracellular liquid, extracellular plasma. So this is transmitter gated channel. Yeah, when there is no transmitter, it is closed. When transmitter binds to the receptor, then there will be exchange of ion. Sodium ion will go through inside, potassium ion will go out. Okay. There's also voltage gated sodium plus ion in the adjacent region. And from here, there will be induced current since there is a potential difference. This is highly positive, this is negative, current will flow here. Similar here, positive, this is negative, current will flow towards here. Okay, is there any questions so far? So if not, that will be all for.
Sir,
Yes.
I want to ask a little bit. So the myelin sheath is acts like a resistor, or
Yes, it will basically. It has a different resistor values compared to if it is unmyelinated. The unmyelinated part still, of course, have resistor here, but it will have different value. Like in the unmyelinated part, the value will be the same on both sides of the membrane. But in the myelinated part, let's say here, it has different value of R. Yeah, in the cytosol, in the inner part of the membrane, and this outer part of the membrane. So that is the difference why in the myelinated and unmyelinated part of the region.
okay, sir.
Anything else?
Thank you.
So,
No, it's some kind of confusing.
yes, this is not